what is a healthy microbiome?
You know, this is a good question. I get asked this quite a lot. What is a healthy microbiome? And, you know, given that everyone has a different microbiome, you know, everyone in this room here, you, me, we all have different microbes. The answer I always give is one that doesn't make you sick. You know?
Interesting.
You asked me to define what is a healthy microbiome. What does it look like? There's such a spectrum in people and what their microbiome looks like, especially down at the species and strain level, you know. It's you know, if it's making you happy, healthy, you're not sick, then for you, that's probably a healthy microbiome, you know.
Beyond that, though, could you go a bit deeper in terms of,
you know,
constituents, functions, presence, absence?
No. You know, that's the thing. I think people will tell you they can. I don't believe you can. I think, you know, there are certain key functions like short-chain fatty acid production, whatever, that these microbes, fine, everyone knows these things are good for you. But, you know, lots of different microbes make short-chain fatty acids. I would argue that, you know, having a decent amount of butyrate producers, butyrate being one of the short-chain fatty acids, is probably a good thing on balance.
Sure.
Other people would argue that butyrate can be bad for you, you know, because it has sort of effects on host cells. So generally, we're having a debate here where there isn't a consensus, and there isn't a consensus for a reason, right? Which is that the microbiome is so complex and has varied so much between people that actually it's been very, very difficult to say, look, if that person has this microbiome configuration, they're definitely not well. If this person has this, they're definitely fine. And it's just been very, very difficult to get to that stage.
But we've identified across a large number of what I'll call kind of case control studies that there are distinct differences between people who have disease and people who are quote unquote healthy.
So I'm going to put a question back to you. What do you mean by distinct?
Distinct. So what I observe, what I've seen a lot of, is most notably and most consistently reductions in diversity, defined by the ecological indices, Shannon, inverse Simpson, all the things that you...
At what point does that become diagnostic?
Well, we'll get to that. We'll get to that. And I'm being interviewed now. Testing me. I see very frequently differences in relative abundance in particular genuses. So, for example, Proteobacteria.
So I agree. If you have that's the one thing I would say. You know, diversity. I mean, that fluctuates. You know, if we have a lot of studies, for example, where we feed dietary fiber to people, and diversity goes down, not up. Right. And you know,
why? How?
Well, I'll tell you why. So you know, if you say you fed resistant starch, okay, I think you talked about this with Harry Flint when he was on. Ruminococcus bromei is a great resistant starch degrader, right? So if you have Ruminococcus bromei in your gut, that will likely respond to eating lots of starch, right? So it will go up.
Okay, I got
and so it becomes 25% of your microbiota, so everything else drops. So your diversity score goes down because this one bug has taken over, right? So diversity being high always equals good. You know, it's one of these. I guess what talk to me, the one thing that hopefully comes across is that, you know, in the microbiota space, simple statements are very rarely always true. You always need that nuance with microbiota. So usually, yes, a higher diversity is associated with health, but in and of itself, it doesn't mean you're definitely healthy.
I got you. No, I fully agree with that. But
the Proteobacteria point is a good point. I think, Proteobacteria, Enterobacteriaceae type opportunistic pathogen type bugs, them sneaking up in abundance is probably not a good thing, right? But there are people who are ostensibly healthy walking around who have, say, 5-10%, and they seem to be all right. You know, now, you know, again, it's not at a point where if you're trying to talk about the microbiome as a clinical feature, where you can go, right, you've got this profile, you've definitely got disease X, we need to treat you with treatment Y. You know, and I don't think you will ever get that level of precision for any disease in microbiome, I'll be honest.
Let's debate that. I'll just share some more insights because I think I have a bit of an unfair advantage here insofar as we profile, I think, more healthy stool samples than maybe anyone else, potentially in the world now, actually, with open biome ceasing to exist, which is I think is pretty cool. And without delving into anything that's, you know, commercially confidential or strictly proprietary or a trade secret, we do see some fascinating trends, like and consistent trends.
I'm going to call you on trend.
Okay. we see some consistent findings across all the people who filter through our process, which arrives at a cohort who are healthy. So without giving too much away, diversity is consistently higher on these indices than people we've sampled elsewhere. The Proteobacteria thing is consistent insofar as we see less than 5% relative abundance pretty much every single time. The fascinating thing, though, is we do see transient carriage of certain things. They appear and they disappear. And again, I can't go into too much detail, but I think in people who are unhealthy, this transient carriage of pathogens or pathobients, can you call them that? Is that a word that we
I
like?
call them opportunistic pathogens, but that's fine.
Yeah. In someone who's maybe not healthy, who maybe has reduced diversity, they can somehow find a way to kind of stick around.
So I think, like I say, like I'm not here to be a contrarian. I really don't like contrarians, honestly. It's contrarianism has gone crazy over the last couple of decades. People just say things just for the sake of it.
Yeah.
I think there's what you're saying there. There's definitely merit in it, right? You know, absolutely. It's well established now across a range of disease conditions. Diversity tends to be down.
Yeah.
Enterobacteriaceae tends to go up. Now, I would ask the question: is that a cause or a result of the disease? And I don't think
that's a tough one.
There, you know, if you have inflammation in your body, inflammation will dampen down the growth of many of the beneficial bugs in your gut. They're more sensitive to it than things like Enterobacteriaceae. And it may just be you've wiped everything else in a little bit, and Enterobacteriaceae takes its chance.
Right.
Now, I do think there is potentially you get this sort of negative feedback loop because they are a little more pro-inflammatory, these Enterobacteriaceae. So I can see how you get in a situation whereby these do a little better because of inflammation. They trigger more inflammation, which means that good bugs never come back to the same level. And so I do think things like that are probably having a role to play.
Absolutely.
In things like inflammatory bowel diseases. I'm not going to argue that point whatsoever. Whether you can take that situation as a diagnostic and say this person X definitely has disease Y, that's what, with the level of precision required for clinical practice, that's where
that remains to be determined. I'll just say one more thing. I think somebody somewhere in the world, and if you're listening and you've got the ability to do this, please do it. I hope we'll show somehow through one of these prospective longitudinal studies that you do get a proteobacteria bloom before the
onset.
That would be so cool. Now,
I agree.
Why it's bloomed, we don't know. But that would be awesome.
I mean, honestly, I agree. I think absolutely. I think longitudinal studies, given the variation in individuals, following people long term, so they almost act like their own control. That is the way forward for the field. Because if you take a group of people X, group of people Y, and just compare them, you will find differences in the microbiome 100%. Just by random chance. It's these longitudinal studies that you're talking about, where you actually get a sort of preceding event that will hopefully lead you to something that's a bit more concrete. So, and the field is doing that. To be fair, I think for 10 years, a lot of people just sampled a bunch of people and went, look, I did this, everyone did it, right? It's what was done. The technology was new. It was the first thing to do was just look, can we see disease X, Y, Z? Do they have different microbiome? But actually that correlation is not necessarily meaningful. And I think the field as a whole needs to move from correlations into mechanisms and actual consistent patterns that, come across the whole repeated study sets.